Okay, hello everyone. Thank you for joining us. Let's go ahead and get started. I'm Julia Willett, editor of the thematic issue on versatile lifestyles of Enterococci as commensals and pathogens in FEMS Microbes. It's my pleasure to welcome you to today's FEMS Microbes webinar on Microbial Dark Matter. The mission of FEMS, the Federation of European Microbiological Societies, is connecting, promoting, and supporting microbiology through knowledge exchange, community building, and creating confluence between people, ideas, and disciplines for a globally sustainable future. FEMS invests in science. We use the income from our journals to fund activities and support our community, such as this webinar series, that enables us to highlight different topics of microbiology and allow you to hear from the scientists that are behind the key research published in FEMS journals. Note that when you as an author publish in FEMS journals, and when you as a reader use these publications, you directly support the various activities in which FEMS invests back into science and builds value for the microbiology community. Our webinar today will focus on cutting-edge studies exploring novel microbial sources for antimicrobial compounds, innovative protein profiling and pathogenic bacteria, and groundbreaking non-invasive techniques for characterizing anaerobic gut fungi. Our aim is to engage and prepare researchers in service to the scholarly community, as well as, of course, to the science that they are working on. Indeed, it's the enthusiasm and dedication of all the microbiologists, FEMS member societies, volunteers, authors, editors, readers, staff, and many more that make FEMS the organization that it is today. On that note, I would like to introduce Kimberly Klein, Editor-in-Chief of FEMS Microbes at the University of Geneva in Switzerland, Laura Chavarria Pizarro at the Instituto Tecnológico de Costa Rica, Katia Núñez Montero, the Universidad Autónoma de Chile, Jeanette Grunwag at the Arctic University of Norway, and Markus Neureder at the Universität Innsbruck in Austria. I want to direct our audience to please present your questions to our authors by using the Q&A function at the bottom of the screen. We will select some questions to ask our authors during the panel discussion at the end of all of the presentations. Kimberly, would you like to say a few words about the topic and FEMS microbes?
Yes, indeed. Thank you, Julia. And welcome to everyone in the audience. It's great to see so many people joining us today. So as Julia said, I've been the co-editor-in-chief of FEMS Microbes since its inception about 5 years ago. And so FEMS Microbes is a gold open access journal. We cover all aspects of microbiology and the microbial world around us. The journal aims to publish basic and applied research within a diversity of areas, including industrial, environmental, clinical, and veterinary microbiology, as well as methodological advancements that are of general interest to a wide audience. One thing in particular we strive to do is to be a venue to early career scientists to read and publish and contribute to our journal. And we do this in a variety of ways, from early career poster prizes at things like conferences to highlighting early career authors through things like Editor Choice Awarded publications, social media amplification, and individual researcher profiles. We also do seminars such as this, or webinars such as this, where we put early career investigators like Julia and early career scientists like our speakers at the forefront. And so I really encourage all of you early career researchers out there that are listening to get in touch with us if you think the journal might be a good fit for your research, or if you have any questions at all. We're always happy to chat about publishing with us at any time. And finally, we host sessions at the annual FEMS Microbes Congress, which is being held in Milan this year in July, where I'm co-chairing with Julia a session on microbial dark matter. And so Julia highlighted a few of the goals of this particular topic with this webinar and this associated session at FEMS Microbes Congress. And so what we're trying to do is really increase awareness of the scale and significance of microbial dark matter and unculturable species and unknown gene functions, to advance insights into methodological approaches to study microbial dark matter, and really to improve the understanding of ecological and biotechnological implications of microbial dark matter. And so we hope that this forum and its associated session at the Congress in Milan will really provide a forward-looking perspective on how to think about and unravel microbial dark matter, and how this can contribute to innovation and microbiology and beyond. So with that, Julia, I hand it back to you.
Great, thanks so much, Kim. So our first talk today will be presented by Laura and Katia, so please take it away.
Good morning in Costa Rica. My name is Laura, and Katia and I are going to present some results related with our research in social wasps in Costa Rica. So, as many of you know, we have a serious problem with the antibiotic resistance because common diseases and minor injuries could become deadly because several antibiotics are no longer effective to treat bacterial infections. So for this reason, it's important to perform research in new sources. So scientific community has been studying defense mechanisms of several organisms like plants, mammals, and invertebrates. And among the invertebrates, many studies has focused in social insects because social insects can establish symbiotic relationships with antimicrobial producing microorganisms. But maybe you can ask, why study social insects? What do we have in common with these organisms? If we analyze the characteristics of the human society and the insect society, we can find some characteristics in common. For example, we both live in closed places when we regulate temperature and humidity. The social insects use the nest and we use buildings or our homes. Also, we have trash inside our homes and buildings, and the social insects, they accumulate the larval feces inside the nest. And this could create an environment that could favor the pathogen development. Also, for example, we share the space with other individuals, for example, in the city and the buildings, and social insects, they share the space with thousands of individuals. Nevertheless, the social insects maintain under control the pathogens along thousands of years because they develop different mechanisms to defend their nest. Why we select Epiponini wasps among the social insects? Because it's a group that have been poorly studied, but also because they present a high diversity of adaptations. For example, they build different types of nests. They also live in different environments and they have different reproductive strategies. Also, they have the brood cell reutilization. As you can see here, the larval feces accumulate under the root cells, so this could create a substrate for parasite development. So the wasps, the workers, have to create some mechanisms to protect the larvae, but also the adults against these pathogens. For this reason, we isolate actinobacteria from cuticle and salivary glands from adult workers in several species of Epiponini. We identify it at molecular level, then we evaluate the inhibitory activity against different pathogens, and also we evaluate the potential as a source of natural products. So we select species from 3 different clades in Epiponini phylogeny to obtain diversity of strategies, and also we want to obtain a diversity of regions. So we collected wasps from 4 regions in Costa Rica, 1 tropical dry forest in Guanacaste, a urban area in Cartago, and 2 lowland forests in the Caribbean and also at South Pacific. We collected 20 individuals for each nest, as you can see in this table, to perform the isolation of actinobacteria. We follow Madden and Hamilton protocols to isolate the actinobacteria. For cuticle, we use a solution of PBS, and for salivary glands, we use a sodium acetate dissolution. Then we identify the actinobacteria by molecular analysis. We extract the DNA using modifying 2 protocols, as you can see here, and we send the DNA to post-imaging to amplifying and sequencing. We perform antimicrobial assays by the top agar method. So we culture the pathogen microorganisms, and then we put small pieces of the actinobacteria cultures to measure the inhibition halos. We selected bacterial species with harbor known human pathogens as the ATCC strain of Escherichia coli, Pseudomonas aeruginosa, and Staphylococcus aureus. And we also use one parasite of insects, Bacillus thuringiensis. Then we obtain the genome of 6 strains that present higher inhibition halos to analyze the biosynthetic gene clusters. And we obtain the genomes by Illumina, and all the details and the protocols we use are in the paper, but you can also ask later in the questions. Also, to confirm the taxonomy assignment and determine the evolutionary relationship between the strains we obtain and the closest taxa, we use a multi-locus phylogenetic analysis, and we follow these steps as you can see here. And then we analyze the functional and biosynthetic genes using an anti-smash for secondary metabolisms, functional annotations. Also, the protocols are in the paper. Okay, we obtain 49 actinobacteria strains. Most of them were identified as Saccharopolyspora, as you can see in the slide, Streptomyces, and Amycolaptosis. But we also obtain strains of other genera like Nocardiopsis, Cocuria, Sucamorella, and Microbacterium. It's important to highlight that this is the first report of Sucamorella for social wasps, and also is the first report of Microbacterium for Epiconini wasps. Also, it was interesting that we obtained strains only from specific regions and also from specific wasps. For example, Sucamorella, we obtained it only from Bolivia nest and from this region, from South Pacific of Golfito. Other strains like Streptomyces, Microbacterium, and Cocuria, we found in different places. For example, we obtained 39 strains from cuticle and 10 from salivary glands. For example, Microbacterium, we obtained only from salivary glands. These other 3 strains we obtained from cuticle, and these others, for example, the Saccharopolyspora, Streptomyces, and Cocuria, we obtain it from both sources. Because several strains presented inhibition against bacteria, 61%, we hypothesize that some of them are in the wasps for protection. For example, 97% of the 61 inhibit strain, they presented activity against Bacillus thuringiensis. So we hypothesize that in other insects like ants, Epiconini wasps use these strains to protection because they have activity, as you can see, against the bacteria, mainly against Bacillus thuringiensis. In the case of the strains that we found in the salivary glands, we also hypothesized that they could be related with a digestive role, because in other insects, the researchers found that these bacteria could be associated with the enzyme production. But our strains also presented activity against the human pathogens. For example, 83% of the strains presented activity against E. coli. Also, 63% present activity against Pseudomonas. And we didn't find a lot of activity against Staphylococcus. And also we found very small halos. But with the other bacteria, Pseudomonas and E. coli, we found activity. And also we found 6 strains that show inhibition against all the test bacteria. For example, Streptomyces, these two strains, Amycolaptosis, and also Saccharopolyspora and Nocardiopsis. So we thought that these strains could have a lot of potential to search for new antibiotic compounds. And now Katia is going to present you the results about the genomic. I'm going to, Katia, can you hear me?
Yes, yes. Hi, everyone. As Lara said, then we selected those strains with most antibacterial activity. So we select these 6 strains that Lara presented in the methodology, and we sequenced the whole genome. 4 of these strains were Saccharopolyspora, and 2 of these strains were Streptomyces. Those were the ones with most antimicrobial activity and with a diverse range of antimicrobial activity. Regarding the genomics results, we have a genomics report about it with all the details on the genome and the annotation. I just want here to mention that the genomes of Saccharopolyspora were about 6 million bases, and the genomes of Streptomyces were 7 and 9 million bases. The next one. With the genome, we started to analyze the phylogenetics. So we make some analysis related to comparisons with other strains of Saccharopolyspora and other strains of Streptomyces. Regarding Saccharopolyspora, by the time we did this study, we found around 26 reference genomes. So we compared with these genomes. With Streptomyces, we select like about a little bit less of 400 genomes to compare. And regarding Saccharopolyspora, we found all or 4 strains from this species, from this genre, sorry, belong to a different clade in this genus. So we compared also through an average nucleotide identity report, and any of the known species of Saccharopolyspora match with ours. So most probably this is, correspond to new species of Saccharopolyspora, but among them they are really related, and they share more than 95% of the genome. So probably they belong to the same species among them, but it's not a reported species. About Streptomyces, I'm not showing the phylogenetic tree because it's like huge, but we found that one of these Streptomyces has no match above the species threshold, that is 7L, but HG, the second species, it's really related to Streptomyces globisporus. And interesting that we compared more than the genome, and we tried to look in the references where this species is coming from. And for Streptomyces globisporus, it was reported in a beetle, and it was related to protection of the beetle because of the production of antimicrobial compounds. So probably new questions will arise from this about the specificity of this species in insects. The next one. Then we conducted the identification of VGCs, of biosynthetical gene cluster, because with this genomic analysis, we also want to evaluate or to look into the genes that might be producing molecules with antimicrobial activity. So our idea was to identify this cluster. We used anti-SMASH, and we found about 17 different types PP, all those that are really related with sorry, I just saw the message that I was gone. I'm here. So we find many types of biosynthetic gene cluster that are related usually with production of antibiotics, and we classify them based on the similarity with known BGCs. So we have some of those that were like 100% complete, others that were not complete, and others that were completely unknown. So studying these and comparing with literature, we can go to the next slide. We select some of those that showed lower similarity because we are interested in what is new. So we found all these compounds that are related compounds produced by these species, but with low similarity. Low similarity means that not all the genes of these biosynthetic gene clusters are present in our strains, but some of these genes might be present in our strains. So some of the genes in the clusters might be related with the production itself of the biosynthesis of this molecule, but some others can be related with regulation and other kinds of stuff that doesn't give us much information about the similarity of a compound. So we look into these comparisons to see which genes are really related with the production in our strains. We can go to the next slide. And comparing and selecting some of those that might be more interested in our case, because we were wondering if some of these BGCs might be related specifically with the insect, with protecting the insect by producing some antimicrobial compounds. And we found these three, and you can see the comparison of the cluster, or in our strains, if the first line, and the cluster, the reference cluster is in the second line. For the first row, we have pyrisidine 8-1. In the second row, we have nystatin, and in the third row, we have selvamycin. And particularly, these three has been reported in other social insects for protection, for antimicrobial protection. For example, pyrisidine A has antibacterial activity and was first reported in ants, and its activities also related to this protection. And selvamycin was reported, is an antifungal molecule, was reported in streptomyces. And after this reporting in streptomyces, some other authors have found it in other species, but not in Saccharopolyspora. Here in this study, we found it in Saccharopolyspora, and as you can see, the cluster, it's the red, the dark red genes are the biosynthetic genes. So in all of these cases, the biosynthetic genes have similarities with the genes in our strains, but they also have differences, and we have additional biosynthetic genes. So maybe these biosynthetic genes will produce something similar, but not the same molecule. But what we want to show here is the potential of these strains for producing probably novel antimicrobial compounds related with the social insects. The next one.
Katia, I don't know if you want to say the conclusions or.
Okay, yeah. So as a conclusion of the whole study, this was the first report of several actinobacteria isolated for Epopony adult wasp with in vitro confirmed antibacterial activity. We demonstrate that actinobacteria associated with social wasp adults have antimicrobial activity and BGCs sharing some of genes related with the production of antimicrobial compounds already tested for insects. And actinobacteria associated with social wasp could be a potential source of novel antibiotics based on our results about BGCs, and also based on the novel nature of this species, because of the selected 5 of the 6 selected bacteria were novel species. So thank you very much for your attention, and we will be happy to take your questions after the presentations.
Okay, thank you.
Okay, thank you so much for that, Laura and Katia. That was fantastic. Our second presenter today will be Jeanette. So Jeanette, please take it away.
Thank you, Julia. And thank you so much for the opportunity to come here and have this talk. Yes, hello everyone. My name is Jeanette, and I'm presenting to you from Tromsø. I hope maybe where you are, there's more spring than here. We have snow still. And I recently defended my PhD. And what I want to talk to you about today is this paper that we published a year ago, which was the first paper of my PhD, where we've been looking at protein profiling of serine hydrolases and penicillin binding proteins in Enterococcus faecium. So what I want to start with is to introduce you to the Enterococci a little bit. So the Enterococci, they are gram-positive bacteria, which means that they have this thick layer of cell wall peptidoglycan outside their cell membrane. And this cell wall is the target of many antibiotics, as we probably many know, and also where, for instance, the penicillin binding proteins are doing their function. So in humans, the Enterococci can account for up to 1% of the normal gut microbiota. And these are primarily represented by the species E. faecium and E. faecalis. And my focus has been on E. faecium. And not only are Enterococci found in humans, but also in a large variety of other terrestrial species. And one of the characteristics of Enterococci are that they are very adaptable and hardy organisms that can survive a wide variety of environments. And this might be part of why they have a lot of intrinsic resistance mechanisms, and also why they are really good at acquiring new resistance mechanisms to antibiotics. And E. faecium is particularly good at that, and is very resistant to many types of antibiotic agents. These are some examples, such as cephalosporins, aminoglycosides, and so on. And two specific examples are aminopenicillins, which include ampicillin, which is actually the preferred antibiotic for treating enterococcal infections. But as we can see in most countries in Europe today, more than 75% of hospital isolates are ampicillin resistant. And then we also have vancomycin resistance in Europe that differs quite a lot from country to country. But in general, the trend is that we see that vancomycin resistance is rising. And that's quite concerning because vancomycin is the last resort drug for difficult enterococcal infections. So this means that the treatment options can be quite limited for enterococcal E. faecium infections. I want to point out that E. faecium exhibits a distinct clade structure, which it clusters into clade A1, A2, and clade B. Clade B has recently been proposed to belong to another species, E. lactis. But interestingly, the potential to cause disease varies among these clades, which is shown here in the golden bars. So the strains within clade A1 are highly associated with infections and the hospital environment, while A2 and B strains are more commensal from various sources. It's also a little bit interesting to note that the clinical A1 strains have, on average, significantly larger genomes than strains from clade A2 and clade B or Eulactis, which is likely due to acquiring genes encoding virulence factors needed to be more virulent and cause infections. But a big portion of the genomes are encoding proteins that we know very little about beyond simple annotations that is often based on homology to proteins in other species. So what we have aimed to do in my project has been to investigate proteins in E. faecium, which are highly uncharacterized, to see if some of these could be potential therapeutic targets, so that we could get some new ways of treating antibiotic-resistant E. faecium. And so to do that, we have used a technique which is called activity-based protein profiling. And ABPP is quite a powerful method that can provide insight into the active proteome of the bacteria. And so this technique utilizes what is called an activity-based probe. And these probes are small molecules which act as enzyme inhibitors. And they are designed specifically to target specific enzyme families. And usually they consist of a reactive group, which will be the component that binds directly to the active site of the enzyme. It's also sometimes called the warhead. And then you have a linker region, which connects the warhead to a reporter tag. And the reporter tag can be a fluorophore for profiling, or, for instance, a biotin molecule for enrichment and further identification. And so the procedure of doing ABPP is generally quite fast and straightforward. So here we have some E. faecium. And what we do is we incubate the live bacteria in a culture with the probe. And of course, the bacteria is expressing different types of enzymes at all times. And then the probe will find and bind to the specific enzymes which are active. And this leads to us having a labeled selection of the protein that we can profile or enrich and identify. So a fluorescent probe can be used with a simple STS page to profile the enzymatic activities. While using a biotinylated probe in conjunction with mass spectrometry analysis, we can get more information like sequence information and identity of the proteins. Yes. So the results of my study, here we have the profiling of the penicillin binding proteins. So we labeled using a probe which is called Bacillin FL, which is specific for PBPs. And we labeled in different strains belonging to the different clades of E. faecium. And we also profiled after growth on 3 different media. So it was LB agar, blood agar, and BHI agar. So in general, we detected bands between 76 and 150 kilodaltons, which corresponds to the 6 known PBPs in E. faecium. And then we also identified a band which was smaller, around 52 kilodaltons, which was not assigned, but which we identified as a D-ala-D-ala carboxypeptidase, which is encoded by DDCP. And this basically suggests that there is a 7th PBP in E. faecium, sorry, supporting also some earlier predictions done in previous publications that there is a 7th PBP. So, sorry for my voice. So we did see that the growth medium had some influence on the PBP activity, mainly that there were less bands detected after growth on LB compared to blood agar and BHI, which blood and BHI agar is more favorable for the Enterococci. And then we also see that in blood agar and BHI, we have a few more bands. This band corresponding to the dark blue arrowhead here, we think is a PBP5, which is an important PBP for ampicillin resistance. Yes, so in general, we see that the growth medium seems to have some effect on the PBP activity. Then we also saw a few clade to clade variations after growth on LB. Mainly that we have this band that represented by the orange arrow. This is then active in the clinical A1 strains and not so much in the commensal A2 and B strains. And we see the opposite for the band corresponding to this green arrow. And then we also see some small variation from strain to strain. For instance, that this strain A1B seems to not be expressing, or at least the enzyme is not active here, and also that this A2 strain is expressing something quite highly here. So at the current moment, we don't really know what this means for the bacteria, if that could have some implication, for instance, for the cell wall structure and composition. But if there were time, it could be interesting questions to look into. Yes. And then we did an analogous experiment to the PBP one, but here we used the FP-TMR probe, which is specifically targeting serine hydrolases. So again, we have the same strains and the same growth conditions. And what we observed was that there was very low overall variation in the activity profiles between the strains and also between different growth media. There were some small variations which we could see maybe that clade A1 strains showed a higher conservation of activity patterns, with only one exception, maybe that this one AB also is not has a lacking band here in LB and BHI. Agar. While in clade A2 there were slightly more variation in the activity profiles. For instance, the band represented here with this pink or purple arrowhead is in some strains absent or less active. In the blood agar, we see that there is one band with high molecular weight, which pops up. But we don't think this is actually related to the bacteria, but rather the blood agar, because we've seen it also in various other bacterial aftergrowth and blood agar. So this conservation that we see of the serine hydrolase activity patterns, especially in clinical clade A1 strains, at least suggested to us that maybe they play important physiological roles in the bacteria in some way. And so we continued the work with the serine hydrolases, and we used the biotinylated version of the probe, and then we could use streptavidin enrichment to pull down the probe labeled proteins and use LC-MS analysis, which compared the enriched proteins to a negative control, which was a no FP biotin treated control. So what we saw was that we could identify with high confidence 11 different serine hydrolases that was largely uncharacterized before. And the size variation was between 22 and 66 kilodaltons, and there were a few different domain types. So there were a few alpha-beta hydrolases. We had one amidase, an esterase, a few patatin-like phospholipases, and some peptidases and proteases. And so several of these serine hydrolases also showed homology to known virulence factors in other bacteria. So for instance, ESH6 and ESH9, we are related to two proteins that we are actually working on in S. aureus, which we think plays a role in stress response. And then you also have these patatin-like phospholipases, which also have been associated with bacterial virulence in other species. And one of the good things is that so 5 of these shared homology with human proteins, so that the rest of the serine hydrolases had no homology to human proteins, which is good if we would want to use them as therapeutic targets. So to summarize, there were some differential expression of the PBPs across the different strains and clades, and this could potentially impact, for instance, some antibiotic resistance. And then we have the serine hydrolases. We saw that they were highly conserved. We know that they are accessible because the probe is able to label them in live cells, and they are functionally active. So it means that they are quite strong candidates for novel antimicrobial targets. And all the 11 serine hydrolases that were identified are validated due to their probe binding ability. So this sort of the paper was the groundwork for further characterization of these serine hydrolases. And I see I don't have much more time, but it's I can sort of hint at that we continued some of the work. So we did a few types of assays with knockout mutants on some of these serine hydrolases, and we could, for instance, see that one of them is important in the survival of these insect larvae. And we also did some work on isolating the enzymes and doing some simple characterization of their activity on different substrates. Yes. So thank you for your attention. I hope to be able to answer some questions afterwards. Okay.
Great. Thank you so much, Jeanette, for sharing your work and those very cool labeling techniques. Our last speaker today will be Marcus. So Marcus, please take it away.
All right. Hello, everyone. I hope you can see my presentation, hear me well. I'm going to speak a little bit today about a paper that we published last year in FEMS Microbes. I also want to start out by saying thank you for inviting me to this webinar, and that I'm very excited to talk about this topic today. And it is going to be about near-infrared spectroscopy and hyperspectral imaging as novel tools to characterize anaerobic gut fungi. Now, what are anaerobic gut fungi? Their name already hints that these organisms are strictly anaerobic, which means they're extremely challenging to cultivate. They are a basal clade of the kingdom of fungi. They're called Neocolemysticomycota, and they actually were only discovered to be fungi in 1975, when a central dogma of microbiology was broken that only oxygen using fungi exist, and these were then discovered to be also fungi. And they currently have 22 described genera, but there's actually believed to be a lot more. Now, this is a webinar on microbial dark matter, and I think the AGF fit perfectly within this because, as you can see on this figure here on the right, which is from the global herbivore microbiome study, you can see that there is a lot of genera that have not been cultured yet, and that there's a lot of new genera that were discovered in this study. So there's a lot of taxa out there that we don't know about yet and that are still to be discovered. And what's also relevant about them is that they're not currently available in any culture collections. So you cannot go to a culture collection and get these organisms. It's really about a tight network of researchers around the globe that sort of send these cultures around between each other to study them. Now, anaerobic gut fungi, as again their name hints, originally inhabit the intestinal tract of herbivorous animals, because this cow that we can see in this picture here is actually not able to digest the plant material that it eats itself. So it relies on a complex consortium of microorganisms to do that for it. And these anaerobic gut fungi are part of their rumen, and they're very potent in breaking down lignocellulosic plant material through mechanic means, through the hyphae, but also through a collection of very potent enzymes. And this is also displayed by the fungi in the bottom picture here growing on the plant material. And this is why they're also relevant for research, is because in biogas plant, similar things happen as in the cow rumen, where organic waste that is often recalcitrant plant material is deposited, and then methane is produced. And if we could yield enzymes from these fungi that help make this process more efficient, that would obviously be very useful. And also in the natural environment, these fungi co-occur with they're commonly associated with methanogenic archaea that are also then present in cultures when they're isolated from the environment, which is usually done through the collection of feces. Now, there's not only challenges with cultivating anaerobic gut fungi, there's also challenges with their genome. They have very large and repetitive genomes, which makes the usual shotgun sequencing approach is quite challenging, and there's very few reference genomes. They have an extremely high AT content, some of the highest known in any living organisms. And what is especially challenging for routine analysis is that they have large length variability in the ITS marker gene region. And there are now approaches with using the large subunit to identify these organisms, but still there is an interest in alternative characterization methods for these understudied and often difficult fungi. And the ones where our paper comes in and what I want to talk about today is the near-infrared spectroscopy and hyperspectral imaging using mid-infrared spectroscopy as alternative characterization methods specifically for these fungi. And now that I talked about what are anaerobic gut fungi, I also briefly want to talk about what is infrared spectroscopy. So the infrared light is in a wavelength region that we can see here is just adjacent to the visible light. There's also different categories of infrared light. We have near infrared, which is called that way because it's near the visible light spectrum, and there's mid-infrared, which is a little bit further away from that. And what is so great about infrared spectroscopy is that, as you can see on the bottom left, we have these infrared waves hitting molecules that start excitations. And these excitations are not only energy and wavelength dependent, they're also concentration dependent, which means this gives us qualitative as well as quantitative information about our samples. And on the bottom right corner, we have a typical infrared spectrum where we have these peaks arising at different wavelengths, and the position of the peak is energy dependent, and the height of the peak is then concentration dependent. And with all of this information, which peaks are present, which wavelengths are absorbed, and also how much is there, we can draw conclusions about samples. And if we record infrared spectra, we sort of get fingerprints of certain samples, and we can distinguish these samples by those fingerprints. And an additional benefit of the spectroscopy method is that it's fast and non-destructive, so samples can be used for further downstream analysis. Now, I also want to briefly talk about hyperspectral imaging, which is an excellent technique that sort of combines the best aspects of microscopy and spectroscopy, where, as in microscopy, we have a spatial XY axis that we can use to investigate our samples. And then this sample is divided into smaller pixels, where each of these pixels a spectrum is recorded of. And we then gain a spectral information of the sample that is also divided into a spatial information. So each little pixel of our sample, we get spectral information from, and can even distinguish within one of these recordings the absorption differences along the sample. Now, the aims of our study were to test the applicability of near-infrared spectroscopy to characterize HEF strains and find out which molecules are responsible for differentiation. We wanted to test whether it's possible to differentiate cultures harvested at different culture ages. And we wanted to test the applicability of hyperspectral imaging to characterize HEF strains and to check for morphological differences within strains. Because of time reasons, I will only talk about points 1 and 3 right now, but if you're interested in the others, you can find that in our publication. Now to the procedure, we grew our strains on defined medium at 39 degrees Celsius for 1 week, then washed and lyophilized these strains, which then eventually yields a powder of fungal samples. You can see one of our anaerobic serum bottles here on the right, because as I said, strict anaerobicity, so no oxygen can get into those cultures. You can imagine how challenging it is to cultivate them using syringes and maintaining the 39 degrees Celsius. We used 3 different strains: Brecoromyces ruminantium, Aneromyces mucronautus, and Cecomyces communis, and we yielded just under 30 samples for each. This is also representative of the challenges of getting pure cultures of these fungi. At our institute, these were the only available pure cultures of anaerobic gut fungi. We then did an NIR measurement of the dry fungal powder, and for the hyperspectral imaging measurements, we actually took biomass out of these serum bottles with a syringe and deposited that on calcium fluoride slides to then be investigated with hyperspectral imaging. Now, this is a short graphical abstract of what we did. You can see here that we have our 3 HEF strains at the top that we recorded NIR and hyperspectral imaging spectra of. We then used multivariate data analysis for model building to then differentiate these strains from one another and also discriminate unknown new strains and say which strain is what species depending on the spectral fingerprint they yield. For the hyperspectral imaging measurement, I also included here a schematic overview where you can see a HEF slide on the top left that we then took a hyperspectral imaging measurement of where we have different pixels that we get spectra of, and then we can combine the spectra of certain regions. So for example, from this round sporangia, we can get a characteristic sporangia spectrum, and we can get a characteristic hyphae spectrum if we combine a spectra from this hyphal region. And then you can see at the bottom that these average spectra are different from one another depending if we scan over hyphae or sporangia. And for differentiating the strains, we obviously generated spectra of these small regions that are representative of hyphae and sporangia, so to get a good overview of the sample. Now, to the results of our measurements, these are the average spectra for the 3 strains that I mentioned, and also chitin as a abundant cell wall molecule in fungi. And as a comparison, you can see it's quite similar. And you can see that these average spectra are different from one another. And I want to highlight region D here in the spectrum, where we have absorption from amide functional groups and also in the center from an OH functional group. And of course, this corresponds to different biomolecules in the biomass of these fungi, where amide correlates with chitin and protein content, and OH absorption correlates with chitin and carbohydrate absorption. And now if we look at our different strains, you can see that we have different absorption maxima. So Cecomyces in red has the highest chitin and carbohydrate absorption. On the other hand, Aneromyces mucronatus has the highest amide absorption. And so not only through different compounds being present, but also through different relative abundances of these functional compounds, functional groups, and biomolecules in the biomass of the fungi, we can differentiate these strains from one another. And if we look into what we did, it is that we did 3 times a random sample split into calibration and validation set. And how this works is that we used 70% of the recorded NIR spectra to build a model. And then we used this model that was based on Mahalanobis discriminant analysis to predict the remaining 30% of unknown AGF strains. And this was done with a 96% prediction accuracy, so it's very precise and robust, this technique, in predicting unknown strains from these AGF cultures. And for hyperspectral imaging, I again show you here the average spectra that are now shown in the mid-infrared region because we used a different region of the infrared spectrum. And again, the average spectra are different for the 3 tested strains. Most relevant is on the right here, the so-called fingerprint region that I now want to zoom in. And what I also want to show you here is that these are individual spectra, so each individual sample is depicted here. And you can see that for different functional groups, we have different absorption. And this is also coherent for individual samples depending on their strain. So the in-strain spectra are much more homogeneous, and there's heterogeneity between the strains, which makes it very easy to discriminate these strains from one another. And we did the same for hyperspectral imaging that we did for NIR, where we split our samples into a calibration and validation set at the ratio of 70 to 30%. And we used 70% of our spectra to build models on Mahalanobis discriminant analysis, and then predicted the remaining 30% of unknown samples with, on average, a 97% prediction accuracy. So also this approach is very precise and robust in detecting, in determining the species of an unknown strain. Now I want to briefly discuss these results. So for the near-infrared spectroscopy method, we have an easy, fast, and accurate method that is independent of molecular approaches. Our technique is non-destructive, so we allow for further downstream analysis. We still have this dried powder. We could still extract DNA from it. We can do whatever we want with it. Now, some of the drawbacks of our study is that we only tested differentiation and genus level on 3 of the 22 known genera. This, as I already mentioned, was mainly an issue with not having more pure cultures available. This could be extended in the future if more pure cultures become available. And the NIR method is only applicable to pure cultures with sufficient biomass because we scan the entire dried fungal biomass. Now, for the HSI results, we have an easy, fast, and accurate method, again, and it is independent of molecular approaches. It is not so non-destructive because we deposit our plates, our samples on calcium fluoride plates, but in theory, we could still use it. It's just a much smaller sample amount, and that's why we also didn't care so much about wasting it. And it was again only tested at genus level on 3 of the 22 genera for the same reasons as mentioned before. And an additional benefit is this method, because of the spatial information that we gain through microscopy, we can scan through small fractions of entire cultures. And actually, if there's a degree of impurity or particular plant matter, as I showed you in the picture before, it would not be as big of an issue because we can select for regions that we know are pure. Some of the future outlooks that I want to mention is for both of these methods, we could have an expansion to more cultures to differentiate species level and more general differentiation. We could include strain of different ages to build more robust models, and we could have implementation of this technique into future novel characterization. So with our proof of concept study, we want to say, well, this is a functioning tool that is working well. And if future strains are characterized, we could employ this to characterize these strains. Now, for hyperspectral imaging, we could try testing cultures with particular plant matter present because we can avoid it. We could try differentiation of strains grown in core culture that we could try to separate microscopically with our spatial information. And we could then, at the end of the day, at some point, hopefully, evaluate the application of this technique to environmental samples where we could differentiate strains. And that would perhaps one day allow for the development of a culture-independent technique. Now, I want to briefly also mention a complementary paper that we have just published a few weeks ago that is based on a very similar procedure on the exact same samples, but it uses MALDI-TOF and DART mass spectrometry for HEF discrimination. And as you can already see in the graphical abstract, the procedure is very similar. And this paper is already out there if you're interested. And it's more from a technical point of view than from a biological point of view. With that, I've already come to the end of my presentation. I want to thank all of you for listening and your patience. I want to thank the people at the HippoF project that helped me with the work from the microbiological point of view. If you're interested in more of this anaerobic gut fungi work, you can visit the website, hippof.com. And I also want to thank the people at the Institute for Analytical and Radiochemistry here in Innsbruck that provided the instruments to do these measurements. And if you're interested in either of our two papers, I briefly mentioned the QR codes here. Thank you. And I'm happy to now start the question session.
Okay, thanks so much, Marcus. What a very, very cool technique. I would like to thank all of the presenters this morning for sharing their work with us. I think now we'll move into the Q&A session. And while everyone's kind of getting ready for that, I want to remind everyone that all of today's speakers have recently published this work in FEMS Microbes. And so please check out the very, very cool papers that go along with the presentations from this morning, as well as all of the other great articles in those issues. So it looks like we have a couple questions for Laura and Katia in the Q&A. So Laura, do you know the function of the ectoine in the strains?
Hi, I can help with that. So thanks for the question to Juliana, I think. It's a very interesting question. Actually, the ectoine is an osmolyte, and it's actually really widespread in bacteria. Not only actinobacteria, in Protobacteria, Firmicutes. In my case, I cannot say it's in all actinobacteria, but we have sequenced many actinobacteria, and it's always there. Ectoine, it's always there. This osmolyte has been related to different stress conditions, not only the water loss, but it can help protecting the biomolecules from UV radiation, from temperature, heating, or frozen. So these wasps were isolated from a tropical environment. Maybe one could imagine that it's not that stressful, but I think every bacteria gets some of this stress, for example, from UV radiation at any point. But the truth is we cannot know what exactly is doing ectoine in these strains, but I think I'm more in the evolutionary genomics, so I think probably this ectoine is in bacteria since many time ago, as a protective osmolyte in biomolecules, and it can be active or not in our strains. And actually, it's really cool to see it as a core biosynthetic gene cluster that we can find in bacteria.
Great, thank you. Okay, there is another question here for Laura and Katia. Has the same kind of symbiosis been seen in other insects living in communities apart from wasps?
Yes, actually, ants have been extensively studied. So they found several species of actinobacteria. Actually, there is a study here in Costa Rica. The selvamicin, the compound that Katia mentioned, the presentation, was isolated from a colony of ants here in Costa Rica in La Selva. Also, we can find this relationship in bees and also in other solitary wasps, in termites. So yeah, solitary wasp also, and yeah, it's like a very common relationship between the insects and the microorganisms. So yes.
Yeah, great. Okay, I actually have a question for Jeanette. I'm curious if any of the hydrolases that you found or that you identified in your study are found on plasmids versus the chromosome, especially in some of your clinical isolates, and whether you think there is potential for some sharing between some of these strains and that contributing to antibiotic resistance.
Yeah, quite interestingly, all of those 11 that we identify are on the chromosome and not plasmids. So, but what we see is that they are very conserved though across different strains. So if they share, it could be that they are not really shared on plasmids. No, definitely not. And what we also see is that they are maybe more physiologically relevant for the virulence rather than very acting as virulent factors themselves.
Sure.
Yeah. I hope, yeah, did it answer something?
Yeah, absolutely. Thank you. Okay, we have a couple questions in the Q&A from Marcus. So the first is it possible to discriminate very similar strains as well?
Yes. So for near-infrared spectroscopy, it has for bacteria been demonstrated that there is discrimination is possible even below the species level. So even strains that belong to the same species can be differentiated. This sort of depends also because it is a biomass using technique. Is there differences in the biomass between strains? And if that is even the case for closely related organisms, then yes, it can be detected. And for anaerobic gut fungi, I was not able to test this, but it would be very interesting to investigate.
Great. So there's another one in here that is maybe related. So can NIR be used for detecting and differentiating strains in a consortium?
So one of the things with NIR is that it scans through the entirety of a sample that you give it. I mean, especially for microorganisms, because usually you yield small amounts of this sample. And if you have a consortium and you scan through this with spectroscopy, you sort of only get an average value of that consortium. You cannot discriminate which organisms are present in this consortium. But if you look at the hyperspectral imaging, where if you can spatially separate different organisms within the consortium and then take representative spectra of these different organisms, then yes, it would be even possible in a consortium to differentiate them. Depending also on the spatial scale, how well can they be spatially separated before you obtain the spectrum?
Sure, sure. Okay, let's see. Well, thank you to everyone who has added questions for speakers to the Q&A. If you still have questions you'd like to be answered, please go ahead and put those in there so we can get to them in the last few minutes of our webinar here.
Julia?
Yes.
Can I ask a question?
Of course.
This, I think it goes maybe both to Marcus and Jeanette. I'm curious about how much the environmental conditions of how you're growing your microbes affect your various outcomes. And then if you can say anything about how you're modeling your growth conditions in vitro, how that might speak to the in vivo condition. And maybe, Marcus, you look primed to go. So I'll let you go first and then maybe come back to Jeanette.
Yeah, sure, I can start. Yeah, it's a really good question, and as I already mentioned, it is a biomass technique. So depending which molecules accumulate in the biomass, it differentiates. And of course, if you use different media, these things can change. If you use we used a very protein limited medium, and if you use a much more protein rich medium, I'm sure these patterns would change. And we also did some investigations that they do change. So if you use NIR, it is critical to use robust protocols to grow these strains. So if you want to apply the model that we built, you would also have to use the same conditions of 1 week of growth, defined media, 39 degrees Celsius. I think this would be required and for this technique, yeah.
Thanks.
Yeah, very interesting question. I think what's quite fun with ABPP is that you can just choose a lot of different environmental conditions that you want to test. So it's not limited to certain media, etc. But in my case, with the, for instance, serine hydrolases, we saw that, well, although we did just test maybe 3 different conditions, that for these specific proteins, the environmental conditions didn't have that big of an effect. So that they are all very active despite the different conditions. And in terms of like the in vitro versus the in vivo, that's also very, of course, interesting. And yeah, there's definitely a jump from saying, okay, it's grown on blood agar, that therefore it will behave like that in the body, in the blood. But there is also possibility to use the ABP method directly in mice. That's been used, so it's not just limited to bacteria, but it's been used in some cancer studies and stuff like that. So you can also definitely use it in vivo as well, which makes it quite, yeah, cool.
Yeah, that's exciting. Cool. Thank you.
Okay, it looks like we have maybe one final question in the Q&A, a sort of technical question for Marcus. So if you think about comparing these results to sort of traditional molecular techniques, can you just quickly comment on sort of accuracy and efficiency of these new techniques?
It's a challenging question to answer because obviously these molecular techniques are also being tried to improve. There's new primers being used that are specific for anaerobic gut fungi. I will say if you try to use just regular fungal ITS primers, your prediction accuracy will be quite low. So I think you will be around 70%. So if you compare it to like the general method, it's much higher. But then with newly designed primers, and also if you target, for example, the LSU instead of the ITS, then I don't know exactly what the prediction accuracies are, but I would assume much higher than if you use non-HEF targeted molecular approaches.
Great. Okay, well, I would like to thank all of our presenters for being here today and sharing these great insights into this fantastic topic of microbial dark matter. Thank you to everyone at FEMS for facilitating this, and we hope that you can join us in Milan at the FEMS meeting for some more presentations and discussions about this great topic.